This fluorescence leakage assay is a simple method for addressing membrane destabilization by biomolecules. Using liposomes encapsulated fluorescent dyes, it can be used to compare different membranes interacting peptides.
This method is especially useful to investigate membrane destabilization by cell-penetrating peptides in order to understand their mechanism of cellular internalization and to assess their involvement in direct membrane translocation.
Compared to biological membranes, liposomes enable control of the phospholipid composition in order to mimic different lipid bilayers such as plasma, endosomal, or mitochondrial membranes.
To prepare LUVs for their use as cell membrane mimics for a fluorescence leakage assay, use a Hamilton glass syringe to mix phosphatidylcholine, sphingomyelin, and cholesterol at a 4:4:2 molar ratio. Use a rotary evaporator to evaporate the methanol chloroform under vacuum for 46 to 50 minutes at 60 degrees Celsius until a dried lipid film has formed. To reconstitute the multilamellar vesicles, resuspend the dried lipid film with 1 milliliter of lipid hydration solution and vortex thoroughly until the dried lipid film has dissolved. After confirming that all of the lipid has been incorporated and that no film is left on the sides of the flask, freeze and thaw the vesicles 5 times for 30 seconds in liquid nitrogen and 2 minutes in a 30-degree water bath per cycle. To prepare lipid extruder, insert 2 Heaps buffer-treated filter.